PacBio is a provider of long-read DNA sequencing. It was founded in 2000 by Steve Turner and Jonas Korlak. It was based on their thesis work when they were both graduate students at Cornell University. PacBio doesn't only manufacture and sell the instruments that produce the sequencing data. We also work with our customers to develop novel bioinformatics tools that bring insights out of these sequencing data. For example, we work with customers to identify novel structural variants associated with rare diseases. We work with customers to develop novel HLA typing algorithms. We work with customers to understand how to annotate novel RNA isoforms in diseases. These tools help customers go from sequencing data to actionable results, whether that's expedited drug discovery process, better HLA typing, being able to treat customers with the proper dosage for medicine, or develop new vaccines.
Anthony Nolan saves lives through stem cells. We find matches for patients who require a donor for blood cancers. Not only do we find matches, we also research the best ways to help increase the chances of finding a match, making that transplant successful, and also now improving the quality of life for the patients after a donation. DNA sequencing is pivotal for HLA typing. The HLA genes are the body's defense system, and we need to accurately identify these in order to match a patient with a donor. We've been fortunate to work with PacBio for over a decade. Their technology allows us to fully sequence HLA genes. This can be pivotal in detecting distal polymorphisms. We know from our own research using PacBio that the importance of high-resolution typing is paramount to patient outcomes. By using PacBio, we have interrogated the HLA sequences to a new level that has shown improved outcomes for our patients.
We were early adopters of PacBio sequencing almost 10 years ago in my research laboratory, but then we subsequently now, given its improvements in variant detection and accuracy, we're now leveraging that research experience and bringing it into the clinical laboratory, given the fact that it can detect so much more variation in the human genome. As soon as I learned about PacBio sequencing and the long reads from the instruments, the first thing I thought of was utilizing that for pharmacogenomics, because that's incredibly important to be able to phase the chromosomes when we do the analysis. There's a lot of enzymes involved in drug metabolism, and the genes that encode those enzymes are highly variable and they're very polymorphic. And that means that in the general population, the sequence is very, very different. And so employing a long-read sequencing technology to actually interrogate those variant sequences in CYP2D6 allows us to provide information on how that person will metabolize many medications.
We have a lot of things we want to work on, and you know, we have concentrated a lot on germline DNA, but the other things that come with that automatically is methylation. And so actually being able to pull in methylation, annotate that at the same time, along with the variants that you get, is I think one place that's going to be, you know, very critical, probably right now, mostly for rare disease too. And then the next one is actually going from being able to do full transcripts. And I think bringing that in, that's going to be a harder problem. We can look at those separately, but actually bringing those all in together to create a full multiomic analysis of an individual is going to be one of the big things about increasing diagnostic yield. And it's just where everybody's going to be going. It's what needs to be done, is actually get a full a measure of everything about a person's genome. That's what the genome should be. It shouldn't be just DNA, germline DNA. It should be all of these things together. The biggest problem we face with aggregating and annotating variants across a large population or a controlled data set, especially when we get into the types of variants that long reads are very good at, with tandem repeats and structural variants, is that you end up with the breakpoints. These events can actually be, I think, much more likely to be recurrent, and you actually find a difficulty of saying that this variant is in this individual is the same one I've seen in this other individual. And that is one of our big next steps that we have to do, is identify how to aggregate these in a way where you say that I've got a variant at a position, I've got 100 people with that variant or some form of that variant. What is the same about them and what is different about them?
What is different between long-read sequencing technology that PacBio provides and other second-generation sequencing technology is the read length and accuracy. Imagine you have a book and you're trying to piece the book together and you only have 200 to 300 words at a time. It is much harder to piece that book together than if you had 10,000 words at the same time. PacBio is that 10,000 word technology. The genome is as complicated as a book. PacBio's long-read technology is able to give you much longer pieces so you can reassemble and understand what the original genome looks like. Where we think PacBio's future lays is that by providing long-read DNA sequencing for high-quality genomes and transcriptomes, PacBio can become a part of improving human health in the global health system.